Hi.
Hello.
Man, Gerald, you're so smart. School is so hard. I was sitting in my biology class yesterday, and my teacher said something about I just don't get it. What does that have to do with biology?
Well, Evelyn, take a seat. You have a lot to learn. First of all, it's pronounced meiosis. Meiosis is a formation of sex cells, a biological process of cell division in eukaryotes used to produce gametes in animals and spores in plants.
How does it work?
It's a two-part process called meiosis 1 and meiosis 2. It results in 4 sex cells with half the number of chromosomes as the starting cell.
Is there something about a prophase in meiosis 1? Is that right?
Yes, that is correct, Evelyn. It's the first phase in meiosis 1. It's also a phase in meiosis 2, but that we will get to later. Before prophase occurs, interphase occurs in the cell. This is when chromosomes replicate to form sister chromatids, which are in an uncondensed form. It kind of looks like spaghetti. Then, prophase 1 can happen. In prophase 1, the chromosomes condense and become visible. They group together to form a tetrad. A tetrad is a grouping of double homologous chromosomes. This is easy to remember because tetra means 4, and you can see there are 4 sister chromatids in a tetrad. Then, spindle fibers form and the centrioles begin to move to opposite sides of the cell. The nuclear membrane breaks down and the nucleosis disappears.
Whoa, that's confusing.
No, it's not. Just look at the drawing.
I guess it makes sense when you look at the drawing. So, then what?
Something else very important happens in prophase 1. This is crossing over. This is when the non-sister chromatids exchange alleles. It's like they are exchanging a piece of themselves. Crossing over is very important because at the end of crossing over, each sister chromatid still has information for the trait carried on that chromosome. Genetic information is swapped, but does not disappear. This is important because it creates genetic diversity. If crossing over did not happen, then there is a good chance that you and your siblings would be genetic clones of each other.
Oh, so that's why my sister has brown hair and I have blonde hair.
But that's not all. There is still much more to meiosis. After crossing over, metaphase 1 occurs. In this phase, the tetrads line up along the metaphase plate, just like this. The tetrads line up randomly, with their sister chromatid pair. This also helps to increase genetic diversity.
Oh, let me guess. Then they get pulled apart?
Yes, Evelyn, good guess. This is called anaphase 1. The homologs separate and begin the move to the opposite sides of the cell. Then, telophase 1 and cytokinesis occur. At the end of this phase, the chromosomes will have been completely separated into 2 daughter cells.
So, is that the end of meiosis 1?
Yes. My, you are a quick learner. Can you guess what happens next?
I'm going to guess prophase 2.
Yes! Gold star for Evelyn. Can you guess what happens?
If it's just like prophase 1, then the nuclear membrane and nucleolus break down and spindle fibers form. Then the centrioles move to the opposite sides of the cell.
Wow! You have a great memory. Or did you cheat and just look at the picture behind you?
Well, maybe a bit of both.
So, let's move on to
Metaphase 2!
Correctamundo! The double chromosomes line up in the middle of the cell on the metaphase plate. Next comes anaphase 2, when the sister chromatids split and begin to move to opposite sides of the cell. And finally, telophase 2 and cytokinesis occur, where the chromosomes fully separate and the cell divides. The nucleosis and the nuclear membrane reform and the spindle fibers break down. The chromosomes uncoil and you have 4 daughter cells, each with half of the number of chromosomes as the original parent cell.
Oh, I think I'm ready to go take my test on meiosis now. It's really simple when you break it down like that.
Yes, yes it is.